Okay, so if I pause share, can you see from your end that yeah, no, I was just saying pause share should also work because they wouldn't be able to see the slides anymore.
They'll go back to you. It's
Okay.
When you pause share, your face will just pop up again.
How about right now I'm pausing? Can I see my slides right? Yeah. Okay, thanks.
You're Luke, right?
So no slide, right?
I'm not sure
So
that.
that's good. And if I unpause it,
And also you shouldn't have anybody online. This is all the other people should be in person.
okay. Yeah, if
If anybody asks us for the Zoom, if anybody today and tomorrow asks you for Zoom link, you
and I pause now. Can you see? Also, you are
it
at
just,
the
it just
previous.
stops. It just stops.
Okay, so I can
It just-
move, but his
Yeah, it
screen
just
doesn't
pauses
move.
in that spot.
Okay. Mhm. So this is good, right? We can use this one.
Did you get any emails saying like more people are gonna be missing now, or just put it like?
If I let me check.
Okay. If anybody starts asking me for the Zoom link, I say no because this is the strictly in person.
Yeah. Okay.
All right, so I'll come back and. Take start class. And I'll come back in a half hour just to see what you guys do.
Should we wait any longer for more students? No? Okay, yeah, let's wait for a few more minutes. Yeah.
So what happened?
No, I think it's fine. Oh, just one second. Let me try to move to a different slide, and I unpause, and your side will show up that. Oh, okay. Oh, yeah. Then I can use this one. Yeah, this one's more direct, I think. Yeah. Yeah, I think it's just at that moment people will just see black screen, so it's not. Yeah, so it's still one recording, right? So I think then you can turn off the camera, the mic. Oh, okay. Okay, I think it's about time. Hi everyone. I'm your new instructor for today's session. So today's session is mainly about the Atlas genomics data, which is part of the EHR data repository. So all of the so we have a lot of samples that we have the so for our patients, we collect like biosamples, and also we collect their blood for sequencing, so we have their genetic data. And all of these patients are also linked to the like electronic health records system that you have been looking at like in the past 2 days, so that you have a lot of phenotype information as well as the genetic data for those patients. Yeah. So maybe I should introduce myself. So I'm Lin Yu. So I'm a postdoc here. So my main research is on to analyze the genetic data right now, mainly in psychiatric disorders. Yeah. So would you mind maybe introduce some of your backgrounds, like do you have any past experience with genetic data before? Yeah. Mm-hmm.
I have used genetic data for
Mm-hmm. Nice. So you have used like you have done a lot of genetic analysis before?
No.
No? Okay, I see. What about you?
I'm sure.
Mm.
For the time being. So yeah, we may need to use this sort of this feature.
Mm-hmm.
That's why I'm just.
Nice, great. Yeah. Yeah.
Rachel, I'm
Yeah.
a PhD student in bio. I used genetic data before, but never matters.
Okay, I see. Yeah. Hi. What about you?
I'm one of the pulmonary critical care fellows. I never used genetic data before, but very interested in pulmonary hypertension, fibrosis. Yeah, I think it's interesting.
Oh, nice. Yeah. Yeah, great. Yeah. So it seems most of you have never used genetic data before. But have you used any like command line environment before? Yeah. Okay. Yeah. So that's good. So to access the actual genetic data, we need to have some knowledge on how to use the command line environment, because all data are stored there. So I think this session of the workshop is mainly to introduce you to like where to find the correct things. But I think exactly how we can analyze those genetic data really belongs to another workshop. So we're not going to go very like in depth into how we can like work with the data. Yeah. Hi. Yeah. Yeah. Hi. I'm a new instructor for today's session. So we're just introducing our background to genetic data. So do you want to say something?
Oh,
Yeah.
I'm Leslie. That someone
Okay. Have you worked with genetic data before? Okay. And do you have any experience in a command line environment? No. Okay. Yeah. Nice. No, you can be here. Yeah. Yeah. We will introduce some of the basic concepts. But I recommend if you want to learn specifically, let's say command line or analyzing genetic data, there are workshops in our collaboratory workshop series that yeah, you can like learn more in depth. Yeah. Mm-hmm. Okay. So yeah, let's go in. So yeah, today I'm going to introduce 3 aspects of the Atlas genomics data. One is from the EHR side, like so there are tables in the EHR system that gives you the IDs, the set of IDs of the patients with biosample or like genetic data specifically. And so I will introduce those tables and how we can combine those tables with other information like the encounter events or like the patient demographic information. And another part I will introduce is the genetic data itself. So the genetic data, as I mentioned, is stored in a Linux system, which is only accessible through a command line interface. So I will introduce where we can find the genetic data in that system. And lastly, I will introduce some of the tools that are available in the Linux system that you can use to work with the data, some very basic applications. And so if there are some specific analysis which require some specific tools that you want to use, then you have to reach out to their like IT guys, so they will need to install those tools for you. It's a very complicated process, but for some basic analysis, they have already installed some softwares for us. Okay, so I just want to introduce what the UCLA Atlas data is. So this whole thing is called a UCLA Atlas Community Health Initiative. So so you can go to that website. They have a lot of introductions. So basically, they recruit all of a lot of patients from multiple UCLA health sites, and they aim to enroll at least 150,000 patients to this UCLA Atlas Biobank to improve precision health. Here I put down a few facts about the Atlas data. So the Atlas, the name Atlas just means like at LA. Yeah, so it's very straightforward. So all of the Atlas samples are linked to the EHR electronic health records, as I mentioned before. So it's a really good way that you can combine the bio like specimens with their phenotype data and also like longitudinal phenotypes. And the goal is to recruit at least 150,000 patients, and the first batch of this EHR-linked genetic data came out around 2020, and at the time there was 28K samples. I think right now there are over 60K samples, and the data that we are going to see today contains about 30 like generic data of around 30K samples. And one of the strengths of the UCLA Atlas like Biobank is that it has really diverse ancestry backgrounds, because they recruit patients from the LA like neighborhoods, and we know it's like LA is a really ancestrally diverse regions. So, and there are actually papers that they publish on this Atlas data that they identify the ancestries of the patients, and they found people from all over the world. And lastly, all of the data are de-identified. So that's why we have so many different IDs for different patients. Like in the EHR system, I'm sure you have already seen there are patient IDs that like you need to use, and also in the genetic data there are sequencing IDs, and there we need to somehow map them together. So so the exact steps of how they identified are really complicated, and I also don't know the full detail. But a few things that they did is that I think they removed some of the extremely young and old people, and they also shifted the time of all of the events. So all of the time that you see in the table are not the actual time it happens. So they're all shifted. Yeah. Okay, so here is the ancestry diversity thing that I mentioned before. So I got this figure from a paper published last year. They plotted out the ancestry composition of the Atlas data. So you can see we have patients data from all of the 5 main ancestry groups, like the East Asia, South Asia, African American, I think South American and European patients. Well, although a majority of the samples are still European, but we do have a lot of like non-European samples here. Yeah. Another thing is that, so we have the EHR data, but the previous like there's previous studies, research have actually can summarize many of the EHR nodes into more like concrete phenotypes that we can work with. So there are over 1,800 phenotypes in the formats of phi codes have been generated. I'm not sure if you know about phi codes, but it's a code that that they use to summarize from multiple codes so that you can have a more concrete definition of the disease. No, I think from billing codes.
Oh,
Yeah, I think they look at the some of the billing codes and they summarize them into summarize multiple billing codes into one phenotypes, something like that.
so
Yeah.
it's like the same data that just. then-
Yes. Yes. It's part of this published work that they summarize into phenotypes. Yeah, so that they also did some of the GWAS. I'm not sure if you like know GWAS. Yeah. Oh, okay.
Like,
Not in the table we see, but I think you can request from the lab. I think. Yeah. Yeah, I think. Yeah. Okay. So now let's get into the EHR side. Oh, hi. Hi, I'm the new instructor for today's session. Yeah. Yeah. So now let's get into the EHR side of this Atlas data. So the Atlas data are within this bundle called Discovery Genomic Integration Desktop bundle. So it integrates the genetic data from the UCLA Atlas Biobank with a lot of EHR data, as I mentioned before, that are from the DDR that you have seen in the past few days. And we have a lot of resources, like tools and resources available. One thing is the Discovery Data Mart, which is like the EHR data, the genomic files I mentioned in the Linux environment, and you also have access to the Linux virtual machine that you can both access the data and like work with the data. Okay, let me pause the share. I think next slide has some sensitive information. Yes, from clinics, yeah. Yeah, through all of the UCLA Health sites, yeah. So yeah, I think in terms of the fecal phenotypes I mentioned, that I think almost all of the samples that we have genetic data have one phenotype, so like one disease, and I forgot the number. I think they say over 99% have at least one disease, and maybe over 60 or something have two diseases, something like that. Yeah. Yeah. Okay. So okay, are everyone like logged on to the SQL system, the EHR system already? Yeah. If not, let's do it. Yeah.
Like a shortcut for the table name. Did you have to do dot shortcut to, was it like UCLA Biobank map? So you have to do like UCLA Biobank map or BBM. I don't know. Well, but you made, so remember you did like PD?
Yes, first you.
I'm trying to use.
Yeah.
I'm about like this one.
You're not using the archive, right?
No.
No, not the archive, not the archive. So the genomics data, so there are many levels of like genetic data. So there are traditionally there are the micro-genotyping data, and there are next generation sequencing data, like more recent, like a whole exome sequencing or whole genome sequencing. So the difference is that the genotyping was done through a array that's kind of pre-designed with a bunch of positions that you want to look at. They are kind of discrete, so you know which positions they look at. But in the whole genome sequencing or the whole exome sequencing, they allow you to kind of continuously go through your genome and sequence each base. So you are able to see some rarer variants in your genome. But unfortunately, the current release of the ATLAS data is the microgenotype data, which means that you only have some of the common variants to look at. If you want to look at some rare variants, you don't have that. But I heard they are generating the whole exome sequencing data, so which might come out soon. And the data that you see right now are not raw data. It's I think it's been through multiple rounds of QC. I think in their name they say QC5, so at least 5 rounds. Like have been applied to those data. So they are high quality data that I see, and you only see those biallelic SNPs, which means that they either have one type of genotype or the other one. And the genetic data also comes in a lot of formats. One of the formats is the VCF format. The other one is called the PLINK format, and the ones that we'll be able to access is the PLINK format. Okay. So the current sample size of the ATLAS genetic data is between 60,000 to 70,000. I think the 2022 release was 60,000, and the newer release, the 2023 release, is the 70,000. And as I mentioned before, in the EHR data, I showed you the table that contains the SNP information. Those SNP positions, so SNP coordinates, are in GRCH 37 version, but actually the data that we see in the Linux VM are in GRCH 38 version. Yeah.